Okay, thank you everyone for coming to this week's research seminar of the museum. This week, we are very pleased to have one of our new Ramon y Cajal Fellows, Rafael Laso-Pérez. So Rafa did his degree in biology at the Complutense here in Madrid, and that included an Erasmus year in Cologne. He then returned to Germany to the Max Planck Institute for Marine Microbiology and the University of Bremen for a master's in marine microbiology, and stayed there to do his PhD on anaerobic degradation of hydrocarbons in archaea. And then he stayed there as well for a postdoc, also in marine microbiology. Rafa then got a Juan de la Cierva Fellowship, which he held at the CNIB, the National Biotechnology Center here in Madrid, before obtaining the Ramon y Cajal to work here in the Biogeochemistry and microbiology, the Microbial Ecology department, working on environmental microbiology, metagenomics, archaea, and anaerobic oxidation of methane. And I think he's going to be talking about some of those things today. So thanks very much, Rafa.
I'm really happy to be here. I'm happy that there are so many people here to listen to my presentation about little bugs. And I hope that I can guide you through their metabolism, their biochemistry. I know that many of you work with animals and plants, and maybe the microbial world is a bit foreign, but I will try to make it simple. I'm sure that I will not succeed, but yeah, I will try. So yeah, today I would like to talk about my scientific career and the ideas that I'm bringing to the museum. I started in January in the Departamento de Biogeoquímica y Ecología Microbiana. And first, I would like to start with the typical picture about my journey. And as Rob said, I did a degree in biology in Complutense University. Afterwards, I don't know why, but I went to the rainy Bremen, a small city in North Germany. Well, I guess because they have an excellent program on marine microbiology, where I did my master and my PhD under the supervision of Ange Boetius and Gunther Wegner. I was not specifically interested in marine microbiology, but about environmental microbiology, because I really think that health microbiology is a bit boring. It's always the same. It's about harming bugs or the microbiota that degrades polysaccharides. Well, environmental microbiology, it's about a lot of different metabolism, a lot of different phyla. I say that in microbiology, in environmental microbiology, we're in the 17th century, in a time of discovery. It's like the time when sailors went on sea and they discovered continents or islands. Nowadays, in microbiology, we are finding completely new phyla, completely new metabolism. So it's incredible because it's a completely new world. Well, I continued there in Bremen for a while. The rain was not enough for me, and I did a postdoc there from 2019 to 2021. Under the supervision of Victoria Orphan. But then in 2021, I said, okay, this is enough. And I came back to Madrid and to the sunny. And basically, I did a postdoc in CNB with the Juan de la Cierva. And since January, I started in the museum. And today, I would like to talk about environmental microbiology. And first, I would like to start with this beautiful image, the Haeckel's Tree of Life for 1866. And you can think that, well, it's really beautiful, it's true, because you see the different lineages. It was the time when evolution was starting to become a prominent idea. And you see Plantae, Protista, and Animalia. But you have to look really, really down to find microbes. Here are Monera. Basically, at that time, microbiology was a field that, well, it was foreign. It was not so important. It didn't play such a big role in ecology, right? The view changed in the 20th century, it started to change, and already we have a more prominent role of microbes with the Whitaker five-kingdom system, in which you can see bacteria like different lineages, and I will say that the space is more shared with other lineages. The critical point was in the 70s when Besse, in this picture from the 90s actually, Besse presented the three-domain system in which you find that Monera has been divided in two domains, bacteria and archaea. And the third domain is Eukarya and has the four kingdoms like, animal, plants, protists, and fungi. And we know now that this is the well, some people will say that this is a two-domain system, but this picture is more accurate with the real diversity, the real biological diversity. It's so accurate, but it's still missing many clades, because if you go into the field and you sequence, you find a huge diversity of phyla. Actually, you are here. This is animals. Actually, these animals are these few branches, while this is archaea, and all of these are bacteria, and these are different candidate phyla irradiation that have been discovered only by sequencing. So the picture is changing a lot. Every time that we sequence, we find new branches. And now it's clear that microbes probably represent the biggest diversity on Earth. But we don't know what most of these microbes are doing. Why? Because basically 99% of microbial species are uncultured. And when we don't have a culture, we cannot say what they are doing physiologically. We cannot do experiments with them. So it's really a big challenge, because we have to use genomics to infer metabolism. And in the case of microbes, it can lead to mistakes. So my main research themes in the different institutes that I have been are unraveling the role of archaea in the anaerobic oxidation of hydrocarbons. I will explain that first. I identify new clades. I try to culture them to understand their metabolism, and I establish some molecular basis for them. Afterwards, I move from the single cell point of view to a more ecosystem level, trying to identify microbial functional traits at the ecosystem level, concretely at the Antarctic microbial bloom. And nowadays, I have a blank page, and I am developing some ideas to do in the museum, and I will talk about them at the end. Starting from my master and PhD, I have to talk first about archaea. For many of you, maybe archaea is something that you left in the biology studies, so I would like to give a quick update. So as I mentioned, archaea is one of the 3 domains of life, and most of the times they have been neglected, even from microbiologists. Why? Because basically we live in a bacterionormative world. Because bacteria dominate everything, they are usually the most prominent organisms, so people think that archaea don't matter. I will say actually that archaea are the LGTB community in the microbial world. And you can think that, well, that's not completely right, but if you look at history, it's quite similar. Until the 70s, they were not existent. Archaea were not described. They were part of bacteria. They were like, no, that doesn't exist. From the 80s and 90s, they were extremophiles. Okay, archaea exist, they live, but they are still only living in hot and dark places. They are extremophiles, okay? So since the 2000s, archaea are everywhere. They're in the ocean, they're in the land, they're even in your guts. So they are really everywhere. But if we want to check the archaea diversity, we have to see the different clades that we have described. This is an ongoing evolutionary description, actually, because every year we describe new clades and these 3 changes. But in general, we will say that there is an important clade called Euryarchaeota. This clade has a prominent role, and what's really interesting for me, because it has a lot to do with methane. And you know that methane is the smallest hydrocarbon. It's basically a carbon with 4 hydrogens. It's an important greenhouse gas. And the relation with archaea over history was mostly related that many organisms, here depicted in blue, are methanogens. Basically, they produce methane. This is the archaea tree, actually, okay? And all these organisms are cultured methanogens. So methanogens that we have in a culture, and we know that they produce methane. How these organisms do methane is, there are 3 pathways. Basically, they all say many of the coenzymes, but there is only one enzyme that is related to all of them, to the 3 pathways. And this enzyme is methyl-coenzyme reductase, MCR. I want that you remember this enzyme. We can neglect the rest of the biochemistry, but I want that you remember this enzyme because I will mention a couple of times during the talk. This MCR, it's catalyzing this step, the reduction of the methyl-coenzyme to methane. And it's a critical enzyme because for a long time, well, actually still, researchers don't know how this process happens. It's really difficult to understand since methane is only, doesn't have any reactive group, doesn't have an oxygen, for example, that produce a different of electronegativity that will make easier the attack for an enzyme. So biochemists have found that the MCR enzyme has a pocket okay, where the methyl-coenzyme entries, and basically other coenzymes enter, and they may pack all together in a really small pocket, so it's really, really small, nothing can fit, and then the reaction happens and methane exits. But this is not exactly known how it happens, but it's clear that the pocket has to be really, really small. This view of microbiology changed at the beginning of this century when some microbiologists found that actually the enzyme can catalyze the opposite reaction. Sometimes the enzyme, instead of producing methane, activates methane and produces methyl-coenzyme. The organisms responsible for this are called anaerobic methanotrophic archaea, or archaea again, in short, ANME, and they actually perform the process called anaerobic oxidation of methane, because basically they oxidize, well, these are the organisms actually, they oxidize methane to CO2. But they cannot do this alone. They live in syntrophy in the environment. They usually are attached to sulfate-reducing bacteria, which are responsible for sulfate reduction. Basically, they oxidize methane, and they transfer the electrons to the bacteria, and the bacteria does sulfate reduction. And they live together in this beautiful consortia. Sorry for the color blind, but the convention is red and green for these organisms. And they live in this beautiful consortium. However, so far I have only talked about methane. Methane is just one hydrocarbon. There are many more. Actually, if you remember from your biochemistry classes, you have alkanes, for example, this butane that has 4 carbons. You have aromatic compounds, benzene, and you have other hydrocarbons. You could think that maybe these hydrocarbons are not abundant in the environment, but if you see the world map, there are many, many environments, especially in the ocean, where these hydrocarbons occur. Basically, they come from deep oil gas reservoirs, and these deep oil gas reservoirs are the source for around half of the crude oil entering into the ocean. So there is a lot of oil in the oceans. What happened in deep sea sediments is basically you have here a crude oil reservoir, and through cracks or fractures, these hydrocarbons diffuse through the sediment. Here you have different microbial communities. In the first centimeters, oxygen can penetrate. But I'm not interested about aerobic organisms, so organisms that use oxygen. But basically, some organisms can use these hydrocarbons here, but oxygen gets depleted. There is no oxygen here. And when oxygen gets depleted in the ocean, sulfate is the thing that microbes use to respire hydrocarbons, because sulfate is quite abundant and can be used as an electron acceptor. And here in this sediment core, in the sediment layers, anaerobic sulfate-dependent oxidation occurs. So when I started my PhD in 2015, this was the overview of anaerobic oxidation of hydrocarbons. Anaerobic because it happens under anoxic conditions, and different kind of hydrocarbons, from methane until long-chain alkanes. Basically, archaea, the ANME archaea, were responsible for the oxidation of methane, and no bacteria was known to oxidize methane. No organism was known to degrade ethane, and basically longer alkanes than 3, for example, propane, butane, until dodecane, hexadecane, and other alkanes were degraded by bacteria. These organisms were using different mechanisms, different enzymatic mechanisms, while bacteria were using a mechanism called fumarate addition. The archaea were using the alkyl-CoM, this mechanism that I mentioned related to the MCR. Remember this enzyme that I mentioned at the beginning. So for me, it was striking that no archaea was degrading any hydrocarbon. So I wanted to study this question. What is the role of archaea in anaerobic degradation of hydrocarbons? So I got some samples from the Guyamas Basin in the Gulf of California. And basically, in this environment, you can see these microbial mats, these beautiful microbial mats, that indicates intense microbial activity below, because basically, sulfate reduction produces sulfite, and this sulfite fuels this mat. So we got some samples from here, from these hydrothermal vents, where there are hydrocarbon-rich fluids, basically methane and other gaseous hydrocarbons. With that, we established a culture in the lab. Basically, we get the sediment. Basically, we get the sediment, we mix with the medium, we make some slurry, and we distribute through bottles that we close to avoid that oxygen penetrates. And then afterwards, we add the corresponding substrate, in our case, butane. We got a beautiful culture here. It's not as beautiful as your animal and plants, but this is what we work with. Basically, butane is the electron donor. That will mean the food source. It's the food that they use. And sulfate is the final electron receptor, is what they respire, so to say. And we see in this culture, basically, that there was growth. Growth is shown when you see that butane is consumed over time, and when you see that sulfide is produced. Sulfide is the product of sulfate respiration. So there was something going on here. We call this culture butane 50 because it grows at 50 degrees. And when we study microscopically, we could see consortia of archaea and bacteria. The archaea in red, the bacteria in green. The bacteria were from a group that we already know called the sulfofervidus, and they are the sulfate-reducing bacteria. So it seems that the bacteria will do the sulfate reduction. The archaea were from a group called Syntropharchaeum, but we didn't know anything about that. Actually, we gave the name. So our hypothesis was that the consortia was performing butane oxidation coupled to sulfide reduction, but we didn't know the molecular mechanisms. To try to understand that, we use omics approaches. Basically, we did metagenomics that I will explain in short. You have a microbial community with different organisms, and basically you extract the DNA, and then you make small fragments to do a library. Then you sequence, and with these reads that you get, you try to assemble them to get longer fragments of the genome. At that moment, it becomes really funny, or I will say it's funny, so to say, to try to put the pieces together and resolve the puzzle. Because basically, all the DNA of these different communities are mixed, so you have to try to guess which fragment, not guess, but use bioinformatic tools to know which fragment belongs to the red organism and which one to the blue. At the end, you get, if you do your work right, you get a metagenome assembled genome that usually resembles the original genomes, although sometimes you have some sort of contamination or is not complete at all. So with that, we were able to get the genomes of Candidatus Syntropharchaeum, or the archaea, and actually we got two genomes from two species, but today we'll talk in general as a genera because both are really similar. What did genome tell us? Well, we wanted to know if they were oxidizing butane to CO2. So for that, we looked first for the pathway that bacteria use to oxidize butane, but we couldn't find it. There was no pathway related to bacteria. Instead, we have an MCR pathway, so this pathway related to methane oxidation. That was weird because actually methane that has nothing to do with these organisms, and two other pathways that are related to process of fatty oxidation and to oxidation of acetyl-CoA. Basically, these were puzzle pieces that we were not sure how to put together. To illustrate how this puzzle became, basically, this is my puzzle when I was a PhD, trying to put all the roots together and trying to guess where the butane was entering. I didn't find the steps from where butane could enter into the different pathways. So I was really puzzled, and I didn't know what to do. I was not finding the right clue. But then at some moment, I thought, wait, it's really weird, but methane should not be here. And if methane should not be here, maybe that's the reason, that that's the piece that is missing. So I tried to study the methanogenesis pathway closer. To remind you, the MCR in animal organisms oxidize methane to methyl-coenzyme. Okay, that's okay. We could check if this happens. This doesn't happen when we try experimentally. And when we check the genome, we found that each of the organisms have 4 copies, 4 genes for encoding MCRs. That was striking because usually you only have 1. So that was something that we should follow. So I did a phylogenetic tree of this enzyme. Basically, I tried to understand the evolutionary relationships. And here I showed you the tree. In blue, you find the MCRs of methanogens, of organisms that produce methane. In red, you have the MCR of methane oxidizers. So you see here that all the methane-related MCRs, or canonical MCRs, are related, are somehow close. And here are the MCRs of my organisms. You see a long branch, and that means a lot of evolutionary divergence. That means that although we have MCRs that can be found with bioinformatic programs according to some models, the MCRs have suffered a lot of evolution, a lot of divergence. So at that moment, I start to wonder, could it be possible that the MCR is the enzyme that is activating butane to butyl-CoM? That was a kind of risky hypothesis. Why? Because basically, methane is 1 carbon and butane has 4 carbons. And as I mentioned before, the enzyme officially cannot fit anything bigger than 1 carbon. So size matters, they were saying, in the biochemistry. But I said, no, size doesn't matter. We can fit something larger for sure. So basically, we wanted to test this hypothesis, and we make a methyl-coenzyme standard. So this a standard of this product that should be the one produced if butane was activated. And when we check for this standard in the culture, we found an exact peak. Basically, with mass spectrometry, we could detect butyl-coenzyme in the culture. And when we remove butane from the culture, the line blue, you don't find this peak. So basically, that was showing us that the MCR of syntrophorchem was activated butane to butyl-coenzyme. So we resolved the first step, and afterwards it was easier to resolve the puzzle. We basically found the different pieces. Step 1, this is still missing. So we were able to have a diagram of how the butane oxidation occurs in these organisms that will show this biochemistry, but we don't have to go through all these steps. But we published this, and that set up the baseline for the future hydrocarbon anaerobic oxidation. What I mean with this is that after our finding of syntrophorchem here and discovering that syntrophorchem can degrade butane and propane, we have found more organisms doing similar process with different hydrocarbons, but using still the alkyl-CoM. So basically, we have found an organism, archaea anaerobically, that can use ethane. We have found an organisms that can degrade long-chain alkanes. In this case, only the tip of the alkane gets into the MCR. It's called methanoliparia. And for a long time, there was a question about these middle steps. And now, actually, fresh from the print, yesterday we published an article showing that there is an organism called alkanophaga that can degrade from C5 until C12. So basically, now it's clear that archaea play a role in hydrocarbon degradation. They are oxidizing all sorts of hydrocarbons from different lengths. And that has evolutionary consequences, because if you have the tree of organisms with methane MCRs, if we add the organisms with non-methane MCRs, and we add those that are not cultured, but we know that from their genomes that they have MCRs, we detect that numerous archaea encode for MCRs. And we have the hypothesis, and it has been proposed, that actually, if all archaea or all phyla of archaea have an MCR, it's possible that the last archaeal common ancestor, Laca, had an MCR at the beginning of the when it appears. So basically, MCR metabolism could be present in the last archaeal common ancestor. I hope that biochemistry was enough for you. I will try to move now to a more ecosystem point of view, and I will talk a bit about my work in the CMB, where I try to identify functional traits in an Arctic environment. So to start, I will first like to show you this video, if I can. Now, in which you can see how the Arctic environments evolve during months. Basically, the Arctic Ocean is an ocean with a polar oceanography in which during winter, the ice covers the ocean and is really dark and cold, while during summer, ice melts and there is ice-free environments, light and less cold. How does that affect to the microbes is something that has been studied for a while. Basically, we know that during the night, the polar night, there are no light, and basically phytoplankton cannot grow. So nutrients accumulate. As a consequence, the prokaryotic community cannot grow neither, because they are usually feeding from phytoplankton. When the night comes, actually, the phytoplankton start to proliferate because they have enough life, and the microbial community proliferates as well. So the objective of my study was to understand the microbial community succession between seasons in an Arctic environment, with a special focus of the nitrogen cycle. For that, we sample in the polar, of course, in the Arctic Ocean, concretely in the Dease Strait here in Canada, concretely between the continent and the Victoria Island. And we have different samples from March that will be considered, so to say, winter, to July, that will be summer. We use metagenomics and metatranscriptomic analysis that I explained before, so I will go now briefly. And basically, we focus on the microbial community, extracting the DNA of the community, obtaining the DNA, and sequencing the DNA, and basically obtaining metagenome assembled genomes, so the genomes of the different organisms, to try to understand process, relationships, and change in abundance. Because we have samples from summer and winter, we could infer a succession of microorganisms over time. First, I would like to introduce from the results the chlorophyll concentration. You can see here how chlorophyll increases over time, basically indicating that a phytoplankton bloom is happening. So algae are proliferating over time. And from this bloom, we obtain the genomes from 176 species, microbial species. That's a lot of work, and that represents really different metabolisms that to study is a bit complicated from an ecosystem point of view. But first, we try to understand how these organisms change in abundance. Here you see genome abundance, metagenomic relative abundance, and the groups that you see here are phyla, microbial phyla. I will lead you through the main clades, basically starting with alpha protobacteria. These protobacteria are really abundant through the whole period. Especially they are abundant in winter, but also in summer. And at the end of the summer, they become super, super abundant, especially one organism. You also have gamma protobacteria that are not so abundant in winter, still considerable, but increase their abundance over time during the middle months of the bloom. And then they decrease dramatically. You have Bacteroidota that these organisms really, really increase with the bloom. They seem to be really favored by the phytoplankton proliferation. And these three clades actually are clades that we know that are proliferate during phytoplankton bloom. So basically, if this is our algae cells, the algae cells secrete polysaccharides, and these polysaccharides are the food source for these microorganisms. So when there is enough food, they proliferate and they feed on the carbohydrates. But what is happening, it's clear that during the summer, during these months, these organisms proliferate. But what is happening during the winter months? Well, basically, in this case, we have Nitrososphaera, this organism that is an archaea, actually. And basically, they have a considerable relative abundance, around 25%, to then decline during the summer. And we have another organism that doesn't seem too important, but actually at the metabolic level, they play a crucial role, that they are really have a significant abundance during the winter months, and they disappear. So I will talk about these two clades afterwards. So what we did is to study the microbial functional trait of nitrogen, and for that we study many genes. You don't have to know all these genes, I will focus on a couple of them, but here you see expression level of the different genes. You can see that there are different magnitudes in the axis, so you cannot compare directly by the size of the bar, and you can see here time represented by colors. And what I want to focus, what I want to pay attention is, these three genes that in this case are dominated expression by Nitrososphaera, and this gene in which the expression is dominated by Nitrospinota. And you see that they seem to have a similar pattern, that the abundance is really high at the end of March. Okay, we have some movement, but it's because it's thrilling. The results are thrilling. But starting with the results, specific results, I will talk first about Nitrososphaera. So Nitrososphaera we know from literature that they are ammonia oxidizing archaea. So they basically use ammonia as a food source, and they oxidize ammonia to nitrite. For that, they use, I will talk about three enzymes. You don't have to know what they are doing exactly, but basically it's a transporter, AMT, and two catalytic enzymes that transport the ammonia to nitrate. When we check the DNA relative abundance, we have this. Basically, this graph shows for each of these genes, the taxa that are affiliated to these genes. So basically, at this sample, in the first sample, around 25%, 30% of the AMT belong to Nitrososphaera. The magenta is Nitrososphaera, while the rest are belonging to other organisms. Because IMT is a transporter that is present not only in Nitrososphaera, it's present in other organisms. What we see here from DNA abundance is that the ANT of Nitrososphaera are really abundant at the beginning of winter and then decrease in abundance. In the case of the catalytic subunit, AMOA actually dominates almost all the time because they are one of the clades that are known to do ammonia oxidation. And near K, they dominate the winter samples and they decrease over time because other organisms have near K. But this is DNA relative abundance. So DNA doesn't mean expression, doesn't mean that these organisms are using these genes. When we can see that using RNA total abundance, so here shown by TPM. And you can see here the dramatic shift from winter to summer. So during winter, although there are many AMTs from other organisms, the ones that are expressing the AMT the most are Nitrososphaera. They are actually dominating the AMT expression. Well, it happens the same for AMOA and NIRK. What does it mean? Basically, it means that during winter, Nitrososphaera is expressing these three genes basically because it's performing ammonia oxidation. It's really, really living on ammonia during winter, the Nitrososphaera. And they, as a final product, they produce nitrate. And that's important for our next organism because Nitrosospinota is the organism that didn't have a high abundance in the environment but was only during winter, are bacteria in this case that can oxidize nitrite to nitrate. They use for that this enzyme called NXR. And we can see from the expression data that actually NXR is highly expressed during the winter months and then completely disappear actually, during the summer months. So these results are indicating that during the winter, we have two organisms that are actually using, in one case, ammonia and then nitrate to then fully oxidize ammonia to nitrate. This is nitrite, this is nitrate. Sorry, chemistry is always difficult. And they are using chemolithotrophy. Basically, chemolithotrophy is inorganic substance as energy source, and because basically there is not so much organic substances. When we have the shift to the summer, this community changes. So basically, Nitrososphaera disappears. Same happens with Nitrospinota. And we have a summer scenario in which phytoplankton proliferates. And now the food source are different carbohydrates that are being used by Bacteroidota, alpha protobacteria, and gamma protobacteria. So we have an ecological succession for an environment in which chemolithotrophy plays a more relevant role to an environment in which chemoorganotrophic bacteria play a more dominant role. So I hope that you could follow this microbial metabolism. And I'm coming to an end now to present some ideas that I have from MNCN for the museum, basically. And basically, I would like to develop an observatory. I have called it ARGOS, Antimicrobial Resistance by Metagenomics Overview in Sewage System. I would like to use metagenomics to sequence different samples from Canal de Isabel II and try to detect antimicrobial resistant genes. This has been done in other places, like, for example, in this study that they presented, and they have seen that these antibiotic-resistant genes are really related to the socioeconomic environment. It's different in poor countries than in developed countries. With that, I would like to identify hotspots, like, for example, hospitals or farming installations that are the source for antibiotics into the sewage system, and finally, try to implement better policies to remove this problem from the sewage system. And for that, I applied to BBVA, to the Red Leonardo. Hopefully, I will get the results in September, maybe with the idea in the future to upscale this project and have a better overview about that. And with that, I would like to finish, and I'm happy to take questions and to get your feedback. Thank you so much.
Thank you very much, Rafa. We have lots of time for questions. Martina?
Okay, very, very nice, very chemical inside your work. But I would like to ask you two questions. The first one is concerning these hydrocarbons, because they are very close to be a plastic or an oleomer. Can you consider to do something with micro and nanoplastic? I know that you will hate me about it because I always talk about plastics, but I am inside this, so I want to know.
You mean degradation?
Yeah.
Of course, it could be possible. Well, possible. hypothetically, there is an idea to upscale to biotechnological applications. The biggest problem is that usually these organisms live with bacteria in consortia, so it's not so easy to culture them. Usually they grow really slow, like 1-2 months of replication time, so it's complicated. But I didn't talk about methanoliparia because I thought I will be short on time, but I have some slides for that. There is a novel organism that we mentioned that can degrade hexadecane, and in this case, these organisms, sorry, it grows, okay, that should not appear, but anyway. Basically, these organisms can degrade different kinds of hydrocarbons and fully oxidize to methane. Disproportionate, actually, it oxidizes to CO2 and reduce methane. So this will be probably the most interesting organism for plastics. The biggest problem with these long hydrocarbons is solubility, because they are not soluble. And if you put a lot of the oil or hydrocarbon, this organism doesn't grow. You have to have an interface, but it's really early stage. This came last year in 2022. So, I mean, I'm not intending to do so because actually the culture is in China. They have the culture in China, but yeah, I think the idea is to upscale for biotechnology in the future.
Okay, the second question is about the climate change, because you are doing this research about this microbiota during winter and during summer, and everything is changing. Do you know something about that, or you study something about that?
So I think in the case of, let me show you the polar ocean. In the case where I sample, the climate change is, so to say, what has been studied is not so affecting because this is a strait. Climate change is more affecting microbiota in the polar environment here, because here there are two currents, one current coming in that is called Atlantic current, and this is the Greenland current. And the Greenland current is really cold and is really different from the Atlantic current, and they have different microorganisms. And it seems that with climate change, it's happening a process called Atlantification, in which the Greenland current is losing strength and is becoming more similar to the Atlantic current. The microbiota is changing. So that's what I know from the environment. Because in general, the Arctic Ocean, the Central Arctic Ocean, has not been studied during winter because ships are difficult to enter. There was an expedition, I think, 2 years ago, well, during COVID, 2021 actually, that they stayed over winter, but results are not still there. They have not come out yet.
Thank you.
Rafa, do you know if this system is limited by nitrogen? Because
here in the system. So I would like to say that we have metadata about nitrogen, but we don't have. So I'm a bit really limited about that. So I don't know.
Okay, I think it's interesting because one of the main differences between the North Pole and the South Pole is that in the South Pole we have no limitation of nitrogen, and in the North Pole we have.
I mean,
so maybe this can be, I don't know, but
they should have ammonia
related with that?
They should have ammonia during winter because there is ammonia oxidation, but I don't know during summer. I think during summer, actually, if you check, okay, this is massive, this expression is massive, but this is a considerable expression of the ammonia transporter. So for sure there is some ammonia happening because of phytoplankton degradation, but we don't know. I don't have the metadata about ammonia. That's a limitation. This data are from 2014, okay. So it's complicated to get them back.
If it's limited, this is very important.
No, for sure.
Okay, they really play a role. Okay.
So there's one question here and then one from the computer.
Thank you. Rafa, nice talk, so thank you. I have a question about anaerobic methane oxidation. Because you mentioned that there's not bacteria that doing this methane, this kind of methane oxidation.
Hydrocarbon.
Okay.
So bacteria don't do methane, they cannot do, there is no bacteria described that's anaerobic.
Anaerobic, but there is
methyl aerobic. Aerobic,
yeah,
yes.
But because they use a couple way or a couple route to the nitrification. They reduce nitrate.
But
And they couple this to a methane oxidation.
But do you think in that case they produce oxygen, intracellular oxygen?
Yes,
There is this cell.
it's not anaerobic, but my question is that you find this kind of bacteria in your
No,
environment?
usually no, you don't have. It's methylomirabilis, no?
Yes.
Right, yes. No, we don't find that.
Okay.
But at the end they use oxygen because they produce
I know.
the oxygen. Yeah.
Yeah, my question is because in caves that there are the environments that I work, there are many of these sequence related to these clades. So I just have question about cultivation strategies or.
I don't know. I think methylomirabilis is culture, right?
Yes.
I think so. So I will have to check the details about cultivation. But I think you have to have it anoxic for sure. And we could try for sure. And I don't know if they use a liquid medium or I guess they use a liquid medium. But we will have to. I will have to check the details.
Okay. Thank you.
Got a question from the chat.
Okay.
What are the opportunities to harness the biotechnological benefits of methanotrophs in methane mitigation?
In methane mitigation, well, I don't think that biotechnological, they cannot be applied to biotechnological. It's more interesting to know that they remove 90% of the methane that is produced in the ocean. So basically, there are many environments where, basically in the deep sea sediments, there is a lot of organic matter, and at the end, methane is produced. And this organism remove 90%. I don't think that we can put these organisms, for example, in cows or in rice fields, because they need to be anoxic. And at the moment that there is oxygen, they stop. And actually, when there is oxygen, we have bacteria, actually, that can oxidize methane really, really well. So I don't think that there is an application for them. The biotechnological application related to this work that maybe is not about nanoplastic degradation is that in the case of ethanoperedens and archaeum, they are consuming ethane. But since they are kind of simple compared to the others, and ethane is a simple hydrocarbon, there is an idea to reverse the pathway. And instead of consume methane, produce ethane. And we will have ethanogenesis. But that's just been suggested, and the need to upscale this project is complete. Yeah.
Okay, thank you. Any more? Yep.
Very nice talk. Thank you very much. In your talk, you mentioned there were some isozymes of MCR. You mentioned 1 up to 4.
Yeah.
Do you have an idea about the differences between them, and if these differences are somehow located in you mentioned one tunnel?
Yes, so this is an old tree. Well, an old tree. This was the tree when we published the article. Now this tree has become larger. So we know that this clade of MCRs seems to degrade long-chain alkanes. When we find an organism that can degrade alkanes with 12 carbons, with 16 carbons, the MCR fits here. So this is a long-chain alkane degradation clade. Then here, there are some MCRs that are related to ethane degradation. So for sure, here is ethane. And then in the middle, we have the ones that can degrade butane, propane, sometimes pentane, but we don't know the difference between them. So there have been several hypotheses that each MCR is related to an alkane, but it doesn't look like according to transcription data. So another hypothesis is that they can use different concentrations. So depending on the environment, if there is a lot of butane, they use one enzyme, and if there is less butane, they use another enzyme, or according to temperature. But further than that, we don't have much idea. Just transcriptomic data, but expression data are always really a bit more difficult to interpret.
And another question is, do you have an idea about how those large hydrocarbons get access into the cells to be degraded?
So we don't have how they get in, no? That's the tricky question, and when we published the paper, the reviewers were asking about that. But they have to get in somehow, I don't know. I mean, the same for the MCR, they don't fit in the MCR, so basically it seems that they only get the tip in, so, and the rest of the change is outside.
Thank you.
Thank you.
No more questions? Okay, well, in that case, thank you very much, Rafa.
Thank you.
And the next seminar is on the 16th of June. Okay.